Pinnularia occurrence. Pinnularia is a freshwater alga. It is found in pond. It is also present on the moist soil. Cell structure, it is unicellular. Its cell is elongated and elliptical. Cell wall is chiefly composed of pectic substances. Silica is impregnated in it. Therefore, their wall becomes very hard. Their wall is composed of two halves called valves. These halves overlap like petri dish. The margins of the two valves are covered by a connecting band called angulum. The two valves with their inner protoplasts are called frustule. The frustule has two views. The surface view is called valve view and band view is called girdle view. The outer larger valve is called epitheca. The smaller inner valve is called hypotheca. The cell is covered by mucilaginous layer. The surface of valve has minute pores or pits. It produces characteristic markings on the surface of valves. These markings are bilaterally symmetrical. The marking-free area is called axial field. The markings are arranged in linear rows. The axial field contains a longitudinal slit called raphe. It has circular thickening called central nodules in the center. Similar thickening also present at the outer ends of raphe. These are called polar nodules. Chloroplasts contain chlorophyll A, C, beta-carotene and diatomine or fucoxanthin pigment. Fucoxanthin gives the alga characteristic color. One or two pyrenoids are present in the chloroplast. But they do not synthesize starch. Their storage compound is oil or chrysolaminaran. Locomotion. Locomotory organs are absent in Pinnularia. However, it moves by characteristic gliding movements. Circulation of the streaming cytoplasm within the raphe causes this movement. Mucilage helps in this gliding. Reproduction. Shocked face vegetative reproduction. It is the most common method of reproduction. It produces daughter cells slightly different in sizes. Vegetative reproduction in diatoms occurs by simple cell division. The protoplast expands. It causes slight separation of epitheca and hypotheca. Nuclear division occurs by mitosis and cell divides into two parts. Each half receives one half of the parent cell. It synthesizes new valve. New valve is fit into the parent valve. Thus new valves are always smaller than the parent halves. Thus one generation gradually becomes smaller in size. It reacted to minimum size. Then its size is restored by auxospore formation. Sexual reproduction. It takes place by auxospore formation. There are two methods of auxospore formation. A. Gamete formation. In some species of Pinnularia, two cells from common parent or from different parents enveloped in a common mucilaginous sheath. The nuclei of both cells divide by meiosis to form four nuclei. Three nuclei disintegrate. The fourth one enlarges. Its protoplast metamorphosed into gamete. The gametes are liberated from the parent frustules. They fuse to form zygote. The zygote enlarges and then forms auxospore. The auxospore secretes new valve and become adult. B. Parthenogenesis. The protoplast of cell secretes a large amount of mucilage. Therefore, two valves are separated from each other. It separates the inner protoplast. The protoplast grows to its maximum size. It secretes silica-rich pectic membrane. This membrane is called prizonium. New valves are secreted inside the prizonium. Nuclear division does not occur. Therefore, auxospores are formed by parthenogenesis. Importance of diatoms. O. Diatomaceous earth. The abundance of diatoms in early geological periods is confirmed by the presence of large deposits of their siliceous frustules. These deposits are termed as diatomaceous earth. O. Diatomite. The diatomaceous earth is mined to obtain a whitish powder called diatomite, which is used as filters in sugar, brewing and medicine industries due to its high absorbent nature. In the manufacture of heat-proof bricks due to fire-proof properties. In the manufacture of water glass due to the presence of silica.